What if education wasn't just about learning? What if it was about igniting a lifelong passion for excellence? Dopamine is more than a name. It's a promise to empower the next generation of medical minds. With a mission to make medical education accessible, affordable, and revolutionary, Dopamine is here to redefine learning for undergraduates. Join us in shaping the future of healthcare, one brilliant mind at a time.
Hello everyone, I am Dr. Kanimozhi Sadasivam, your mentor for physiology here at Dopamine. I am very happy, excited and thrilled to be a part of your learning experience here at Dopamine. As I already told, I am your physiology faculty. So what is this physiology? What are we going to learn in physiology? As you all know, physiology is a very fascinating subject that deals with the functioning, normal functioning of every part of the human body, right from the cell to the organ systems. You should have a very deep understanding of these physiological concepts, because unless and until you know these physiological concepts, you will not be able to understand the mechanism of action of a drug, or the pathogenesis of a disease process, or the clinical presentation of a particular disease, and also the treatment strategies that we apply. To understand all these things, physiology forms the backbone. So it is right to say that physiology is the backbone of medicine. And here at Dopamine, I would help you understand this fascinating subject in a very easy way, so that the subject is interesting, you're able to grasp it better, and also you will be able to score well in your exams. Now, how did I end up becoming a physiologist? Why I love this subject so much? Okay, so after completing my undergraduate studies from the Lady Hardinge Medical College, New Delhi, I started my physiology journey at the Vallabhbhai Patel Chest Institute, New Delhi. We all know this is a very institute where Professor Arthur Singh Painter identified the J receptors and many more. Physiology, as I already told, is a very fascinating subject. From an academic standpoint, if you look at physiology, I enjoy reading the physiological concepts and I equally enjoy teaching it to my students. From a researcher standpoint, the research options in physiology is unlimited. We all know that even in the Nobel Prize is given under the heading Nobel Prize in Physiology or Medicine. Taking physiology as my career option was the biggest decision and the best decision I did in my life, because not only as an academician, not only as a researcher, even because of the fixed working hours in physiology, I am still able to spend time with my family as well as pursue my hobbies. This fascinating subject always keeps me intrigued and also engaged. Hello everyone. The topic for discussion today is gastrointestinal hormone. This comes under the competency number PY 4.5. So what are the learning objectives? This is a very simple topic. At the end of this discussion, you should be able to classify and name the various GI hormones. You should be able to classify and name the various GI hormones. Where are they secreted from? What are the functions of these hormones? And what are the mechanisms that regulate these? How are these hormones regulated? Okay. The term hormone was defined. Many people have given definitions for hormone. In the year 1935, Huxley defined hormone as an information transferring molecule. Hormone is being defined by Huxley as an information transferring molecule. Information transferring molecule, that is the essential function of which is to transfer information from one cell to the another for the good of that particular cell population. Okay, so in simple terms, if I have to summarize in one line, hormones are messenger molecules which carry information from one cell to the another cell. The first hormone to be discovered was secretin. So secretin is important. The first hormone to be discovered was secretin, and our gastrointestinal tract is the largest endocrine organ of the human body. GIT is the largest endocrine gland of the human body. What was our topic? GI hormones. That means now we saw the definition of hormone. Now what is a GI hormone? What is a gastrointestinal hormone? Along its entire length, the gastrointestinal tract So this, if you see the gastrointestinal tract starting from the stomach along its entire length, it is embedded with endocrine cells that secretes various hormones. So the hormones are the messenger substance that is secreted by the endocrine cells of the gastrointestinal tract. These substances are called as gastrointestinal hormone. Though these endocrine cells are dispersed all along the length of the GI tract, they are maximally present in the mucosa of the stomach and small intestine. So that is why in this picture you are seeing stomach and small intestine. Maximum concentration of the endocrine cells of the GI tract, where are they present? They are present in the mucosa of the stomach and small intestine. Okay. So now these hormones that are secreted by these cells will first come into the general circulation, then from the portal circulation it will enter into the general circulation and then reach the target organs and bring about its function. Okay. Now the cells that secretes these hormones are called as enteroendocrine cells. Why it is called enteroendocrine cells? Because they are present in the GIT. That is why enteroendocrine cells, they secrete hormones. So these are basically endocrine cells. Then we also have another group of cell population called as enterochromaffin. When you see the word entero, it means it pertains to the gastrointestinal tract. Enterochromaffin cells basically secretes serotonin. Then we also have another population of cells called as APUD cells, amine precursor uptake decarboxylation cells. They basically secretes amine as well as polypeptides. Okay. So when it comes to GI hormone, 3 group of cell populations are concerned with it. The enteroendocrine cells, which basically secretes GI hormones like gastrin and others, gastrin, secretin, etc. Then we have the enterochromaffin cells, which secretes serotonin, and we have the APUD, amine precursor uptake decarboxylation cells, that secretes amine and polypeptides. Okay. Now, where I told these cells are located all along the GI tract, mainly in your mucosa of the stomach as well as small intestine. Where in the mucosa of the stomach and small intestine are these cells located? If we remember your functional anatomy of the GI tract, we know the mucosal layer lining, the layer is thrown into multiple folds, right? The mucosal layer of the GI tract is thrown into multiple folds. Okay. Now, projecting from these multiple folds are projections called as villi. Especially when we are talking about small intestine, you have multiple projections called as villi. Between these villi, so here what you see is a single villi. Okay. So between these villi, now we are discussing the location of these different cells. These cells are basically in the stomach, these cells are located in the gastric glands. In the intestine, these cells are located in the crypts of Lieberkühn. Okay, so what you see here is the stomach. What you see here is the small intestine. So if we look at the stomach is also thrown into multiple folds. Okay, in between these multiples, for example, it is thrown into multiple folds. Okay, so in between these folds, it is getting invaginated. Okay, this is the region of invagination. So if we enlarge an invagination and see, what you see here is an invagination. Okay, this in the stomach mucosa lining, there are invagination. These invaginations are called as gastric pit. So what is a gastric pit? Invaginations in the stomach mucosa. Deep down in the gastric pit, you see something called as gastric glands. Okay, so gastric glands are present in the gastric pit. Okay, now these gastric glands are made up of, can you see different colored cells, different types of cells. So gastric glands are not made up of a single cell, they are lined by different types of cells. Okay, namely the blue cell, what you see here, this color is just for your understanding. Okay, so these gastric glands are made up of different types of cells. So one group of cells are called as G cells. Okay, G cells is an enteroendocrine. Just now I told you, enteroendocrine cells. So G cell is an enteroendocrine cell that secretes gastrin. Similarly, we also have another type of cell in the gastric gland which is called as a chief cell. This secretes pepsinogen and gastric lipase. Then we also have another cell called parietal cell. The parietal cell secretes intrinsic factor and the hydrochloric acid. Okay, so all these are enteroendocrine cells. Fine. So now again to simplify, the gastric mucosa is thrown into multiple folds. And when it is thrown into multiple folds, they have invaginations. Okay. These invaginations in the mucosal lining are called as gastric pits. Deep down the gastric pit, you can see a region called as gastric gland. So, where is gastric gland present? In the gastric pit. These gastric glands are made up of different types of cells, namely enteroendocrine cells like G cells that secretes gastrin, chief cells that secretes pepsinogen and gastric lipase, parietal cell that secretes intrinsic factor and hydrochloric acid, and then in the neck of the gastric gland, we have mucous neck cell that secretes mucin, and also we have surface mucus cell that secretes mucin. What is the difference between, it's a very important MCQ question, what is the difference between surface mucus cell and mucous neck cell? See, the mucus secreting cell at the surface of the gastric pit is called as surface mucus cell. The mucus secreting cell that is present in the neck of the gastric pit is called as mucous neck cell. Both of them secrete mucus, but the secretion is alkaline when it comes to surface mucus cell, and the secretion is acidic when it comes to mucous neck cell. That is the difference between both these cells. Okay, so that is the gastric gland, which contains these various enteroendocrine cells that secretes various GI hormones. Similarly, the same type of setting is also seen in the small intestine. So, the equivalent of a gastric gland in the small intestine is called as crypt of Leberquin. Okay, so please don't confuse. Crypt of Leberquin is similar to the gastric gland when it is present in invaginations. Okay, so when the invagination is present in the stomach lining, we call it as gastric gland. When the same type of invagination is present in the small intestine, we call them at crypt of Leberquin. Okay, now where are these crypt of Leberquin? Usually you have these villi, projections from the cells in the small intestine. Okay, why do you need these projections? And on top of these projections, you have, on top of these projections, you have microvilli. So these projections are called as lacteals or villi. On top of them, we have microvilli. All these projections are going to basically increase the surface area of absorption. Now, between these lacteals, you have these invaginations. Can you see? These invaginations are between these villi. Okay, so these invaginations are called as crypt of Leberquin. Similar to these cells, these crypt of Leberquins are also lined by different types of cells which are secreting these various gastrointestinal hormones. As we go through, we will see what are all the cells and hormones secreted. Another fact I want you to concentrate here is, it is richly supplied by blood vessels, and the tip of these gastric gland or the crypt of Leberquin is in close contact with the capillaries, so that the secretions easily enter the bloodstream. Okay, so with this background functional anatomy, let us move on to discuss how the hormones are released. Now you know what are all the cells that secrete these different kinds of hormone and where are they located. Fine. Now we will move on to discussing how these hormones are released. For these hormones to be released, either a nerve stimulation in form of vagus nerve is needed. Okay, vagal nerve stimulation is going to increase the secretions of these GI hormones, or distension of the stomach or distension of the small intestine due to the contents. We eat the food reaches the stomach. So that is causing mechanical distension of the stomach. The chyme is reaching the small intestine. That is again going to cause mechanical distension of the small intestine. The flowchart what you see here is for the stomach. Okay, gastric secretions. So now that is why here we are talking only about stomach, but the same thing applies to small intestine as well. So the food particles in the stomach or chyme in the duodenum is going to cause distension of that respective portion of the GI tract. So distension is again going to cause secretion of these hormones. And finally, the chemical composition of the food we take, whether the food is rich in protein. If the food is rich in protein, again the stimulation of G cells is going to be more. Okay, depending upon the chemical composition of the food, the hormones are also going to be secreted. When you eat a carbohydrate rich meal, the composition is going to be different. Okay, the hormones that are going to be secreted are different. When you eat a protein rich food, then the hormones that are going to be secreted are different. Okay, right? So that is with the how the hormones are released. Okay, so either the mechanical distension of the respective segments of this GI tract, or products of digestion, especially products of protein digestion in the form of peptides and amino acid, or a vagal stimulation. All this is going to stimulate the, let us talk about stomach, is going to stimulate the stomach, especially the G cells. Just now we saw, no? So this is the G cells in the gastric glands. So now any of all these three is going to cause the secretion of activation of G cells. Once G cell is getting stimulated, it will secrete its hormone gastrin. Okay. Now this gastrin will act on the parietal cells in the stomach, because the function of gastrin is to cause HCl secretion. So this gastrin will act on the parietal cell of the stomach, causes HCl secretion. Okay. So that is the mechanism by which these hormones are released. Okay. Now we were moving on to the next part of our discussion. How are we going to classify these hormones? So when you get an essay question on this topic, how are you supposed to approach it? First you will write definition of what is a hormone and what do we mean by gastrointestinal hormone. So that is the first point you will write in your essay question. Then you will write how are these hormones secreted. Okay. Then we are going to write what are all the different portion of the gut. That is, in the stomach, it is the gastric glands. In the small intestine, it's the crypt of Leberkin, which contains these cells, which is going to secrete hormones. Which cell is going to secrete what hormone? So this is what you will write. Following which, we will move on to the classification of the hormones. Broadly, hormones, GI hormones are classified into three types. One is the gastrin family hormone, the secretin family hormone, and the rest will come under others. We are more concerned with gastrin family and secretin family. The gastrin family hormones are, as the name indicates, easy to remember, gastrin and cholecystokinin. The secretin family are secretin, GIP, VIP, glucagon and glycantin. Okay. GIP is gastric inhibitory peptide, VIP is vasoactive intestinal peptide. So under secretin family, you have secretin, we have GIP, we have VIP, glucagon, glycantin. The rest of them like Motilin, substance P, somatostatin, neuropeptide Y, pancreatic polypeptide, guanylin and GRP, gastrin releasing peptide. All this doesn't fit into any of the gastrin or secretin family, so they are put into others. Okay. So that is with regards to classification of GI hormones. Okay. Now we will move and see each of these hormones in detail. In your examination, each of these hormones can come as a separate short note. Okay. So first we will see gastrin. Most important among all this is gastrin. Okay. So already just now we have been discussing, right? Now you can recall and tell me. Gastrin is secreted by which cells? Very easy to remember. G. Gastrin is secreted by the G cells. Okay. So first is, what is the cell that is secreting it? Gastrin is secreted by the G cell. Where are these G cells present? In the antrum of the stomach. Can you see? This is the antral region of the stomach. So the G cells are secreted in the antrum of the stomach and also the duodenal mucosa. So this is the duodenal mucosa. Here also you have G cells. So the G cells are present both in the antrum of the stomach as well as in the duodenal mucosa. What are these G cells? What you see here is a G cell. Okay. How is the cell? How is the, you can, just by looking at the cell, you can describe it. So these are flask shaped cells. These G cells are flask shaped cells with a broad base. These are flask shaped cells with a broad base and a narrow apex. Broad base and a narrow apex. On top of the apex, what do you see? What are these things? These are nothing but microvilli. What is the use of these microvilli? These microvilli has receptors which is going to detect the changes in the gastric content. Is it rich in proteins? Is it rich in carbs? Is it rich in carbohydrates or fat? So that is detected by this microvilli. Okay. So that is with the G cells. Fine. So what did you see? What are G cells? Very briefly, its structural anatomy and then where are they located. Now we will see what is getting secreted from this G cells. So basically, what is the G cell secreting? G cell secrete gastrin. So what? Gastrin is a gastrointestinal hormone. What is the chemical nature of this hormone? This is a peptide hormone, protein in nature. Okay. Now we have many types of gastrin have been identified, but for us important ones are these three types of gastrin. Okay. Now how are the names given to this gastrin? depending upon the number of amino acids, depending upon the number of amino acid sequence, we have mini gastrin. Okay, mini gastrin has 14. It is also called as G14 because it has 14 amino acid sequence. Mini gastrin is less active. Following it, we have little gastrin, which is G17. 90% of the antral gastrin. And what do I mean by antral gastrin? The gastrin that is released from the G cells of the stomach. So 90% of the gastrin that is released from the stomach are of which nature? G17. So this is the most active form of gastrin is little gastrin. And then third, finally we have the big gastrin, which is having 34 amino acid sequence. So we call it as G34. Okay. Usually this form of gastrin is released after a meal and this is not as active as the little gastrin. Okay. So three types of gastrins are there: little gastrin, big gastrin and mini gastrin. G14, G17 and G34. Among these three, which is the most active? G17. Where is it secreted from? Secreted from the G cells of the stomach. And where are they located? Even in the stomach, in the antral region. Now we will move on to the stimuli. Okay. Half-life. After a meal, the antral G17 is released and this provides the most potent stimuli for acid secretion. Basically, what is the function of this gastrin? This gastrin that is released from the G cell will go and act on the parietal cell, which is located close by. And it will stimulate the parietal cell to produce HCl, that is the stomach acid. Okay. The half-life of this gastrin, usually G14 and 17 is around 2 to 3 minutes and for G34 it is around 15 minutes. Where is this gastrin inactivated? It is primarily inactivated in the kidneys and in the small intestine. Main location, it is present in the gastric antrum and the duodenal mucosa. Other than this, where else can you find gastrin? This is important from the MCQ point of view. So, gastrin can also be present. Gastrin secreting cells can also be present in the pancreatic islets during the fetal life. Anterior intermediate lobes of pituitary, in the hypothalamus as well as in the medulla oblongata. And also gastrin is secreted from the vagus as well as sciatic nerve. So these are the extra location other than the gastrointestinal tract. Okay, let us move on to the stimuli. Okay, what are all these stimuli? First, let us look at the cephalic phase. Sight, smell, thought of food. Okay, sight, smell, thought of food is going to cause release of gastrin from the stomach. Okay, or the presence of food in the mouth. The presence of food in the mouth, that will also cause the release of gastrin. Okay, so the sight, smell and thought of food is conditioned reflex. Example for conditioned reflex, already you know. So the moment you think of your favorite food like biryani or whatever is it, what is going to happen? We are going to salivate. Similarly, gastric secretion, the gastrin secretion from the stomach is going to increase. This is the anticipatory increase in the gastrin secretion even before the food has actually reached the mouth. That is example of conditioned reflex. Then unconditioned reflex. The moment you put the food into your mouth, whether you want or not, gastrin secretion will start. Why? Because same anticipatory, the food is going to come into the stomach and we have to, HCl is needed for the process of digestion. So the first, for HCl production, we need gastrin. So immediately gastrin will be released from the G cells of the stomach. Okay. Second is, as we already saw, mechanical stretching of the stomach because of the food is again going to trigger the release of gastrin. Similarly, when the contents from the stomach goes into the small intestine, same way stretching of the small intestine, especially the duodenum, will again cause the release of gastrin. Okay. Clear? So these are all the different stimuli for release of gastrin. Okay. Then we also have the chemical composition, the peptides and amino acids, especially what are the amino acids? Phenylalanine and tryptophan. Phenylalanine and tryptophan are going to stimulate the G cell to release gastrin. Neural, already I told you, vagal discharge via the gastrin releasing peptide is also going to cause vagal discharge through the release of GRP, gastrin releasing peptide, which activates the G cell to release gastrin. All this will cause stimulation of the G cells to release gastrin. Additionally, Blood-borne factors like calcium and epinephrine will also have the same stimulatory effect on the G cells. Those are the stimuli that causes the secretion of gastrin from the G cell. Now, what are all the stimuli that inhibits the secretion of gastrin? In the lumen, acidic content and the somatostatin is going to inhibit the secretion of gastrin. Why acidic content? It makes sense, right? Ideal role of gastrin is what? To produce HCl. Now, when enough HCl is produced, once the contents become acidic, gastrin stimulation, gastrin release is inhibited. Somatostatin is an universal inhibitor, so it is going to inhibit the G cells so that gastrin is not synthesized. Coming to blood-borne factors, anything that is released from the small intestine, easier to remember, anything that is released from the small intestine, secretin, gastric inhibitory peptide, name itself is telling gastric inhibitory peptide, vasoactive inhibitory peptide, glucagon, calcitonin. All these blood-borne hormones are also going to inhibit the G cells from producing gastrin. Okay, now that we have seen what are the cells that secrete gastrin, where is it located, what are all the stimuli, and what are all the inhibitory factors for gastrin synthesis. Now, we will look at the actions of gastrin. Okay. When we are looking at the actions of gastrin, by now I have been repeatedly telling you, which is the most important function of gastrin? Stimulation of gastric acid production. How it produces gastric acid production? By activating the parietal cell. So, gastrin activates the parietal cell. If you remember this in picture, you can see. So, here is the G cells which secretes gastrin. Now, this gastrin that is secreted from the G cell will go and act on this parietal cell. Okay. So, it will go and act on this parietal cell, and when it acts on the parietal cell is going to cause release of hydrochloric acid. Okay. So, thus gastrin causes release of HCl, and also it causes release of pepsin as well. Okay. Then it also has trophic action. Gastrin has trophic action on the stomach and small intestine as well as large intestine. That means gastrin has overall trophic action on the entire GI tract. What do we mean by the word trophic action? When I say trophic action, it means it stimulates the growth of the mucosal lining. Okay. It stimulates gastric motility. Why should gastrin stimulate gastric motility? Okay. Why should gastrin stimulate gastric motility? Because only when the gastric motility is increased, what will happen? The contents are going to get mixed out. That basically it causes churning action of the contents in the stomach, so that the food particles are broken down into smaller substances. It stimulates insulin secretion. It stimulates insulin secretion after a protein meal. It also causes contraction of the gastroesophageal sphincter. Where is the gastroesophageal sphincter located? At the lower end of the esophageal. Basically, it causes contraction of the lower esophageal sphincter. Why should this happen? Then only what will happen? Once the lower esophageal sphincter remains contracted, when the gastric motility increases, the contents from the stomach will not regurgitate into the esophagus. So, that is why gastrin causes contraction of the gastroesophageal sphincter. Similarly, it also stimulates insulin secretion after a protein meal. So, once you have a protein-rich meal, what is going to happen? End products are amino acids that are synthesized as a result of a protein meal. These amino acids will be utilized by insulin for building up of muscle mass. It also stimulates pancreatic secretion. Gastrin also stimulates pancreatic secretion, which is rich in enzymes that aids in the process of digestion and absorption. It increases the antral motility, whereas it prolongs gastric emptying. I want you to concentrate on this line. You should know the difference between motility and emptying. Okay. When I say it increases antral motility, it is referring to the same function as it increases gastric motility. So that proper mixing of the contents in the stomach happens along with the HCl. And also physical breakdown or churning action is also taking place. That is with respect to motility function of your gastrin. It is prolonging gastric emptying. Why should it, when I say it, gastrin prolongs gastric emptying, what do I mean? It is increasing the time the contents remain in the stomach. Okay, so that it has a beneficial role. What is the physiological reason behind this delaying of gastric emptying, prolonging of gastric emptying? Then only all the stomach contents will not be emptied into the small intestine at one go. Because of prolonging the gastric emptying in a graded manner, the chyme will be released so that we don't overburden the small intestine, so that the proper absorption of all the nutrients takes place. Okay, then it also stimulates the release of calcitonin, which causes negative feedback inhibition, right? Inhibitory factors. So that is the summary of all the functions of gastrin. Now, to summarize our discussion or to end our discussion on gastrin, let us wrap it up by looking at the clinical applications. First is gastrinoma, also known as Zollinger-Ellison syndrome. Gastrinoma is nothing but a tumor of the pancreas as well as in the duodenum that secretes gastrin. Okay. Now, this gastrin release is unrelated to feeding. Okay. Irrespective of food, secretion of gastrin is going to be very high and spontaneous. So, once the gastrin is going to be very high, what is it going to do? Increase in HCl production. So, hypergastrinemia is going to lead to increased secretion of gastric acid. Okay. Increased secretion of gastric acid is going to cause what? Gastric ulcers. Okay. That completes our discussion on gastrin. Okay. Now, we will move on to the next gastrointestinal hormone, cholecystokinin pancreatozymin, CCK-PZ. Okay. Why is it called as CCK-PZ? Because this cholecystokinin causes the release of pancreatic enzyme. These pancreatic enzymes are stored in the zymogen granules, right? So, it causes the release of pancreatic juice that is rich in pancreatic enzymes. That is why it is called as CCK-PZ. This is also a polypeptide hormone. Cholecystokinin is synthesized from the eye cells of this upper small intestine. There is an easy way. Small intestine stands for SI. I cell secretes CCK-PZ. Okay. Now, S cells, S stands for secretin. Okay. So, the S cells will secrete secretin, I cells will secrete cholecystokinin pancreatozymin. Gastrin, CCK-PZ both belongs to gastrin family of hormones. So, cholecystokinin is secreted by the I cells of the upper small intestine. Acid, peptides, amino acids and fatty acids containing more than 10 carbon atoms are going to stimulate the production of cholecystokinin from I cells. The half-life of cholecystokinin is 5 minutes. Okay. That means for CCK-PZ secretion, what are the stimuli? Acidic chyme, peptides and amino acid in the chyme, fatty acids, okay, containing more than 10 carbon atoms. All this are going to stimulate the cholecystokinin secretion from the upper small intestine. Now, different types, similar to gastrin, we have different types of cholecystokinin as well. Why remembering this is important for your MCQs? CCK58, again, how are these number givens? Based on the amino acid sequence. Number of amino acid sequences, okay. So, CCK58, CCK39 and CCK33. 58, 39 and 33 is found in the brain. 12 and 8 is only mainly for our interest. 12 and 8 is found in the duodenum and jejunum. CCK4 is mainly secreted from the enteric nervous system as well as the pancreatic nerves. So, this is with respect to different types of gastrin where they are found. Now, coming to the functions of gastrin. Sorry, it is not gastrin, cholecystokinin. Now, coming to the functions of cholecystokinin. Okay. Cholecystokinin causes the contraction of gallbladder. Okay. Where is it secreted? Cholecystokinin is secreted by the eye cells of the small intestine. Now, it causes contraction of the gallbladder. Okay. Why contraction of gallbladder is important? Then only the bile will be released from the gallbladder, which helps in digestion and absorption of fat. Okay. When the gallbladder contracts, bile will be released. The released bile has to enter into the second part of the duodenum. So, automatically cholecystokinin relaxes the sphincter of Oddi, because it is via this sphincter only the contents from the gallbladder is going to enter into the second part of duodenum. It also causes secretion of pancreatic juice rich in enzymes. Already I told you, right? It augments the action of secretin, that is CCK augments the action of secretin. It helps. It is like a symbiotic relationship. Cholecystokinin is supporting secretin. Secretin will support cholecystokinin. It inhibits gastric emptying. Anything that is going to be released from the small intestine will go and inhibit the stomach. Easy to remember. Again, same way, the way we discussed in gastrin secretion, only when the gastric emptying is inhibited and controlled, the small intestine will be able to effectively digest and absorb all the nutrients. It has a trophic action on pancreas. Cholecystokinin has trophic effect on pancreas. It increases the secretion of enterokinase. Enterokinase is an enzyme that is secreted by the mucosal cells of the duodenum, and this enterokinase is important for activation of trypsinogen to trypsin, and this enterokinase secretion is increased by cholecystokinin. It also, cholecystokinin may enhance the motility of the small intestine and colon, so that the contents in the small intestine, once it is completely digested and absorbed, it is moving further down into the small intestine. Along with secretin, it augments the contraction of pyloric sphincter. Gastrin causes contraction of the lower esophageal sphincter so that the contents from the stomach don't go into the esophagus. Similarly, this cholecystokinin along with secretin causes the contraction of the pyloric sphincter so that the contents from the duodenum will not enter into the stomach, will not regurgitate into the stomach. Finally, cholecystokinin and gastrin stimulate the release of glucagon. Okay, so these are all the functions of cholecystokinin and the physiological basis for these functions. Okay, now let us look at the regulation of secretion of cholecystokinin. Okay, in case of gastrin, what did we see? It was a negative feedback inhibition. Only when it comes to cholecystokinin, the take home point what I want you to remember here in this slide is it is a positive feedback mechanism. The regulation is via a positive feedback mechanism. So how does this happen? Contact of the small intestinal mucosa with fatty acids and peptide will stimulate the I cells that causes release of cholecystokinin. Secretion of cholecystokinin will cause contraction of the gallbladder to release bile and also pancreas to release. What will pancreas release? Pancreatic juice that is rich in pancreatic enzymes. Okay, as a result of which what will happen? Digestion of protein and fat will happen. As a result of digestion of protein and fat, what will you get? End products of fat and protein digestion are fatty acids and smaller peptides and amino acid. The smaller peptides and amino acid has a positive feedback effect on the I cells, which keeps on increasing the secretion of CCK. Then when will this process be terminated? When all the fat and protein in that food is digested, and when these contents move on to the next portion of the GI tract, this will be terminated. So that is with respect to regulation of secretion of cholecystokinin. Secretion of cholecystokinin. Okay, what are the other sites where you can see CCK-PZ? It is found in the nerves in the distal ileum and colon. In the brain, it is located in the cerebral cortex. What are the receptors for cholecystokinin? Two types of receptors are there, CCK-A and CCK-B. A is located both in the periphery as well as in the brain. CCK-B, B stands for brain, is located exclusively in the brain. Okay, what is its mechanism of action? It activates phospholipase C, increasing the formation of second messengers like IP3 and diacylglycerol. So that finishes our discussion on cholecystokinin pancreatiosome. Now we are moving on to the third hormone, which is secretin. Secretin comes under the family of so when we discuss hormone classification, we have gastrin family hormones, which is gastrin and cholecystokinin. Then we have secretin family hormones, where the first hormone to be discussed is secretin. As already told, first discovered by Starling and Bayliss in the year 1902. It is secreted by the S cells of the upper small intestine. Half-life is around 5 minutes and it is having a amino acid sequence of around 27. Okay, what are the stimulus for secretion of secretin? Stimulus for secretion of secretin, a low pH, that is an acidic pH, and products of protein digestion. If you remember cholecystokinin, both products of protein and fat digestion, but when it comes to secretin, it is only the products of protein digestion. Now coming to the functions of secretin. It causes secretion of bicarbonate. Okay, that means basically Secretin causes the production of pancreatic juice that is watery and alkaline. So pancreatic juice that is watery and alkaline is produced by secretin. Pancreatic juice that is rich in enzyme. So pancreatic juice that is rich in enzymes will be produced due to CCK. Whereas pancreatic juice that is watery, alkaline and rich in electrolytes is produced by secretin. Okay. CCK is produced by the I cells of the small intestine. Secretin is synthesized by the S cells of the small intestine. Okay. Now, other than this, what are the other functions of secretin? Secretin stimulates the hepatic bile secretion. As already told, it augments the action of cholecystokinin, decreases gastric acid secretion and causes contraction of pyloric sphincter. You know why. It inhibits gastric motility. We already discussed what is the physiological reason behind inhibiting the gastric motility, so that we don't overburden the small intestine. It stimulates insulin secretion. Okay. And it stimulates the small intestinal secretion as well. That is, increases the secretion of succus entericus. So, how is it regulated? Contact of the intestinal mucosa with the acidic chyme and peptides is going to stimulate the S cells, which causes secretion of secretin. Now, stimulation of the once secretin is synthesized, what is going to happen? Synthesized and secreted. This secretin is going to stimulate the pancreas to produce pancreatic juice rich in water and electrolytes. These water and electrolytes is going to flood the duodenum and cause the neutralization of the acidic chyme that has come from the stomach. So, ultimately what is going to happen? The duodenal pH is decreased, right? This acts as a negative feedback inhibition. Okay. So, decreased duodenal pH acts as a, that is, the acid is getting neutralized. Once the acid is neutralized, it is going to inhibit the stimulation of S cells. So, when does the secretion finally stop? Once all the acid in the duodenum is neutralized, the secretion of secretin will stop. Okay. That is with respect to regulation of secretin. Now, let us move on to the next hormone, gastric inhibitory peptide. Okay. So, among all the GI hormones, short notes are mostly expected from gastrin, cholecystokinin and secretin. So, you are supposed to, when you get this question in the exam, I would suggest you can follow the same pattern. What type of hormone is it? What are the cells that is secreting this hormone? How is it regulated? What are all the functions? Okay. And if you can add a short note on the applied aspect, it will be well and good. Okay. Now, let us move on to the rest of these hormones. Now, rest of these hormones, what is important from exam point of view? MCQ questions, Viva questions. Okay. Let us look at gastric inhibitory peptide, GIP. So, GIP is secreted from which cells? K cells of the duodenum and jejunum. So far, you only knew two cells, S cells, I cells. Now, the third cell what we are introducing is the K cells, which are located in the duodenum and jejunum. They secrete gastric inhibitory peptide, which is a 42 amino acid peptide. The secretion is stimulated by glucose and fat in the duodenum. Gastric inhibitory peptide, the secretion of gastric inhibitory peptide is stimulated by the presence of glucose and fat in the duodenum. So, this is the gastric lumen where you have glucose. This glucose is going to, this glucose molecule is going to stimulate this cell. What you see here is a K cell in the small intestine. So, glucose in the lumen is going to stimulate the K cells to synthesize gastric inhibitory peptide. Okay. Similarly, the glucose will also stimulate the large intestine, the L cells which is present in the large intestine, to secrete GLP-1. Okay. Glucagon-like peptide 1. These L cells are also can be found in small intestine as well. So, what happens here is, what is the function? Now, we already saw what is gastric inhibitory peptide, what are the cells that secretes gastric inhibitory peptide. Okay. So, what is the cell? Let us, since we are discussing gastric inhibitory peptide, let's stick on to K cells. Okay. So, gastric inhibitory peptide is secreted by the K cells in the small intestine. Okay. The stimuli for secretion of gastric inhibitory peptide for K cells is the glucose and fat in the luminal contents. Now, what are all the function of this gastric inhibitory peptide? As the name indicates, it is going to inhibit the gastric secretion and motility. Hence the name gastric inhibitory peptide. So, most important function of gastric inhibitory peptide is inhibition of gastric secretion and inhibition of gastric motility. Other than that, it also stimulates insulin secretion. Okay, very important point. They will ask you in Viva as well. What is the role of gastric inhibitory peptide with respect to insulin secretion? Depending upon the glucose load, the amount of gastric inhibitory peptide will also vary, and depending on the glucose load, the gastric inhibitory peptide is going to stimulate insulin secretion. It has a direct correlation. Greater the glucose load, greater will be the insulin secretion. What is it basically doing? It is preparing for the gut to absorb the glucose. That is why this is also called as glucose dependent insulinotropic hormone, GIT. GIP, gastric inhibitory peptide, is also known as glucose dependent insulinotropic hormone. Clear? So, what is the use of this hormone? When the glucose load is high, this glucose dependent insulinotropic hormone, or also known as gastric inhibitory peptide, is going to secrete equal amount, in the sense the same greater quantities of insulin. Why insulin is needed? Insulin is going to cause peripheral utilization of that glucose. So, for glucose to be absorbed, you need insulin. That is why it is causing this secretion. Okay. Such hormones that increase the insulin secretions are called as incretin. What is it called as? Incretins. Examples of incretins are gastric inhibitory peptide, or also known as glucose dependent insulinotropic hormone. Another hormone is GLP, glucagon-like peptide 1. Both GIP and GLP are called as incretins, which causes the release of insulin, Okay, once insulin is secretion, what it will be do? The insulin will cause peripheral utilization of the glucose. Now, moving on to the next hormone, vasoactive intestinal polypeptide, found in the nerves in the GIT. It contains 28 amino acids, half-life is around 2 minutes. Other than the enteric nerves, it is also found in the brain and many autonomic nerves. What are the actions of VIP? Vasoactive intestinal peptide, right? It stimulates intestinal secretion of electrolytes and water. Vasoactive intestinal peptide stimulates the intestinal secretion of electrolytes and water. It relaxes the intestinal smooth muscle, including the sphincters. So, if the sphincters are relaxed, what will happen? The contents are going to go easily into the next segment. It causes dilatation of the peripheral blood vessels. Once it causes dilatation of the peripheral blood vessels, it will cause decrease in blood pressure. Vasodilation leading to decrease in drop in blood pressure. It inhibits gastric acid secretion. As I already told, easier way of remembering is all the contents, most of the contents from the small intestine will inhibit the secretions of the stomach. So, it inhibits gastric acid secretion. It stimulates pancreatic secretions, which is rich in both enzymes and bicarbonate. So, those are the functions of vasoactive intestinal peptide. Now, there is a disorder called as VIPoma, vasoactive intestinal peptide secreting tumor. Basically, it is a pancreatic islet cell tumor where large amounts of vasoactive intestinal peptide is secreted. Okay. Now, what it will cause because of this? It is going to, when we looked at the function, it stimulates intestinal secretion of electrolytes and water. So, from the enterocytes, water and electrolytes will be secreted into the intestinal lumen. Additionally, it is also relaxing the sphincters. So, now when secreted in large quantity, what is going to happen? Large amount of fluid is present in the intestinal lumen, and the sphincters are all relaxed. So, what will happen? Copious diarrhea will be there. Okay, this leads to copious watery diarrhea, which is also called as pancreatic diarrhea. VIPoma causes pancreatic diarrhea, or another name for this condition is Werner-Morrison syndrome. Okay. So in VIPoma, what do you see? Excessive loss of electrolytes and water in the form of watery diarrhea. Moving on to the next hormone, Motilin. Okay. Motilin is secreted by the enterochromaffin cells as well as the Mo cells, which is present in the MO. Motilin is secreted from the Mo cells. Okay. Where are these Mo cells located? In the stomach, small intestine and the colon. So all along the GI tract. What is its function? Before going into the function, how does it act? It acts on the G protein coupled receptors. Okay. It acts by activating G protein coupled receptors. Now what are all the functions of Motilin? As the name indicates, Motilin, it is going to cause motility of the stomach and small intestine. What is important, what is the catch here is, Motilin causes motility of stomach and intestine during the interdigestive phase. What do you mean by interdigestive phase? Interdigestive phase is that phase between each meal. We have, that is the phase between breakfast and lunch, or from lunch and dinner. So the phase where you don't eat anything is called as the interdigestive phase. So Motilin is secreted during this interdigestive phase. Okay. So what it will do? Occurrence of migrating motor complexes. That means MMC, migratory motor complexes, happen because of Motilin. Motilin is secreted by the Mo cells that are present all along the length of your GI tract. Now, what is the use of this migratory motor complexes or the movements that happen during the interdigestive phase? The movements that happen during the interdigestive phase is not for digestion and absorption. It is for clearing of the food residues of that previous meal, so that you are preparing the gut freshly for the next meal. So, Motilin is the most important regulator of migratory motor complexes. Now, coming to the applied aspects related to Motilin, a well-known antibiotic, erythromycin. Okay, erythromycin brings about its mechanism of action by binding to the Motilin receptors. So, once they bind to the Motilin receptor, what is it going to cause? Erythromycin. Erythromycin is going to increase the gastric motility as well as the small intestinal motility. So, where will this be useful? This will be useful in treating patients who have decreased gastrointestinal motility. In all those patients who have decreased gastrointestinal motility, erythromycin could be a drug of choice to increase the motility. Okay. Now, moving on to the next hormone, somatostatin. Okay. Now, somatostatin is secreted by the D cells. If you remember the gastric glands which I showed at the starting of this discussion, we saw the D cells also present. So, these D cells are located close to the G cells. Here you can see. We have the G cells. What does the G cell secrete? Yes, gastrin. So, D cells, what does the D cell secrete? D cell secretes somatostatin. Okay. This G cells are located, D cells are located very close to the G cells. Okay. And two types of somatostatin are there, somatostatin 14 and 28. What are the stimulus for release of somatostatin from the D cell? Low pH in the stomach, that is increase in the acidity of the stomach, and sympathetic stimulation is going to cause release of somatostatin. Acidic pH and sympathetic stimulation will stimulate the D cell to release somatostatin. What is the somatostatin going to do? The somatostatin inhibits the acid secretion from the parietal cell. That is why they are located close to each other. You see, D cell will release somatostatin. This somatostatin is going to inhibit the parietal cells. Okay, not the G cells, the parietal cell. It is going to inhibit the parietal cells from secreting acid. Okay. It also inhibits the secretion of vasoactive intestinal peptide, gastric inhibitory peptide, secretin and motilin. Okay. But most important action is inhibiting the parietal cell from secreting acid. How it inhibits the parietal cell from secreting acid? Either by directly acting on the parietal cells and decreasing the HCl production, or since they are located close to each other, this D cell will go and inhibit G cell. So, when G cell is inhibited, no gastrin. When no gastrin, no HCl. Okay. So, somatostatin can directly inhibit the parietal cell and decrease the acid production, or it can indirectly inhibit the G cell, decrease gastrin, therefore decrease HCl production. Okay, both ways. What else it inhibits? It also inhibits the pancreatic exocrine secretion, that is a pancreatic juice that is rich in enzymes and bicarbonate. It inhibits gastric motility, it inhibits gallbladder contraction, it inhibits the absorption of glucose, amino acids and triglyceride. Basically, somatostatin is an universal inhibitor. Just remember this, somatostatin is an universal inhibitor. Then moving on to the next hormone, peptide YY. It is a 36 amino acid sequence hormone secreted by the small intestine cells in the small intestine and colon. Okay, it is really it is stimulated by fat. Please remember, polypeptide YY, the chemical stimuli is fat. What is the function? It inhibits gastric acid secretion and it inhibits gastric motility and decreases the food intake. So, when I say it decreases food intake, it is an example of anorexogenic hormone. It is an anorexogenic hormone. Very important for your MCQ. Polypeptide YY is an anorexogenic hormone. When we say anorexogenic, means it inhibits food intake. Ghrelin. Next, it is a 28 amino acid polypeptide. You would have heard a lot about ghrelin, right? It is primarily secreted in the stomach. It is an orexogenic hormone. That means just the opposite of polypeptide YY is an anorexogenic hormone. It decreases food intake. Ghrelin increases food intake. Okay. That means in well-fed state, ghrelin levels are less. And when you are fasting, ghrelin levels are more. So it stimulates you to eat more. Then comes guanylin. This is also a polypeptide hormone containing around 15 amino acid. Guanylin is secreted by the Paneth cells that are located in the crypts of labor queen. Okay. What is the mechanism of action of this guanylin? It acts by stimulating the activity of guanyl cyclic GMP. That is why it is called as guanylin. Okay. So, cyclic GMP, once the second messengers get activated, what is it going to do? It is going to cause activity of chloride channel and it increases chloride secretion into the intestine. This is the most important thing. What you have to remember with respect to guanylin also has come as some short notes, and it is a very favorite topic for multiple choice question. Okay, guanylin is secreted by the Paneth cells. It acts via cyclic GMP and it increases the activity of chloride channels. By increasing the activity of chloride channel, what is happening? Chloride ion is secreted into the intestinal lumen. So already I said, since chloride ion are secreted into the intestinal lumen, what is going to happen? Fluid is going to move. When chloride is moving, fluid is also going to move into the intestinal lumen. The receptors for guanylin is present in kidneys, livers and also in the female reproductive tract. What is the applied physiology here? Why are we giving so much of importance to guanylin? Because the enterotoxin that is produced by certain strains of E. coli, we know E. coli causes diarrhea. How does it cause diarrhea? By producing certain enterotoxins which have structural similarity. The toxins that is produced by this E. coli has structural similarity to guanylin. So now what will happen? Once it has structural similarity to guanylin, it is going to activate the guanylin receptor, which is going to cause chloride and fluid secretion into the intestine. That is going to cause watery diarrhea. Okay, so that completes our discussion on the major GI hormones. Now for the other hormones, first is neurotensin. It is produced in the neurons as well as in the mucosa of the ileum, stimulated by fatty acids, and it inhibits GI motility. Substance P is synthesized both in the endocrinal and the nerve cells of GIT. Substance P increases the motility of the small intestine. Enteroglucagon. Glucagon is very similar to the glucagon that is synthesized from the endocrine part of the pancreas, islets. Since it is secreted from the L cells of the small intestine, we call it as enteroglucagon. It is a potent insulin releaser. Similar to our gastric inhibitory peptide, even the centroglucagon is a potent insulin releaser. Okay. Then we have GRP, gastrin releasing peptide, which increases the gastric acid secretion, which is present in the vagal nerve endings, and they usually terminate on the G cells. Okay. So that completes our overall discussion on the different gastrointestinal hormone, their secretion, the cells that secrete them, their function, regulation, and the relevant clinical conditions that applies to each of these hormones. Now, as a part of our revision, let us quickly look at the clinical reasoning questions. So here we have a 10-year-old boy who consumes cheeseburger, fries and chocolate shake. So he is eating a junk food, right? He is basically eating junk food. Now this meal is stimulating the release of several gastrointestinal hormones. Okay, so this meal what he ate is stimulating several gastrointestinal hormones. The presence of fat, carbs and protein in the duodenum will stimulate the release of which of these following hormones from duodenal mucosa. The question might look lengthy, but if you read it again, you will be able to understand what they are asking. Basically, this young boy eats a highly processed food. Okay, this food contains fats, carbohydrate as well as proteins. And it is going to, because of this chemical composition of the food, a lot of GI hormones are released. The question here is, identify that GI hormone which is released, identify that GI hormone that is released when fat, carbs and protein, all these three are present. That is, even fat, carbs and protein acts as stimuli for the release of which of the following GI hormone. Is it CCK, or is it glucose dependent insulinotropic polypeptide, or is it gastrin, motilin or secretin? Which of these hormones? What is the answer? It is the glucose dependent insulinotropic peptide or GIP, gastric inhibitory peptide. Both are the same. How? Let us look at the explanation. See, this GLIP is the only GI hormone that is released by all three major foodstuffs. Please note this point. GLP or gastric inhibitory peptide is the only GI hormone that is released by all the three major foodstuffs, that is carbs, proteins and fat. Now looking at the other option, cholecystokinin. Cholecystokinin will be released in the presence of fats and proteins but not carbohydrates. For release of gastrin, most important is protein in the stomach. Okay. Then for motilin and secretin, it is again mainly the presence of protein or carbohydrate but not fat. So when we think about fat, protein and carbs, it is only the GLIP or GIP. Next we have some multiple choice questions which are all clinical based questions. Okay. We have a 45-year-old man who presents with multiple episodes of watery diarrhea and dehydration. Laboratory tests reveal hypokalemia and metabolic acidosis. So this patient is losing water and electrolytes. Okay. Because of this, there is hypokalemia. Imaging reveals a pancreatic tumor. Which hormone is most likely responsible for this condition? Which is the hormone? As we already discussed, it is nothing but VIP or the vasoactive intestinal peptide. What is the patient suffering from? Pancreatic cholera. It is also called as pancreatic cholera. What is VIP going to do? VIP is going to cause profuse watery diarrhea, okay, leading to hypokalemia and metabolic acidosis. Now we have another 50-year-old woman who is presenting with persistent epigastric pain and multiple peptic ulcers that is resistant to proton pump inhibitors. So we have a patient with epigastric pain not responding to the proton pump inhibitor. When we look at the serum gastrin levels, it is elevated. Okay, what is the most likely diagnosis? So here we have a patient of peptic ulcer not responding to treatment. Serum gastrin level is increased. What is the diagnosis? Yes, the answer is Zollinger-Ellison syndrome, or also known as gastrinoma. Why not peptic ulcer disease or achlorhydria? Achlorhydria and celiac disease you can rule out easily. The confusion will come between Zollinger-Ellison syndrome and peptic ulcer disease. Why? Peptic ulcer disease will usually respond to proton pump inhibitor and the serum gastrin levels will not be elevated so much. Okay? Now, so as we already discussed, Zollinger-Ellison syndrome or gastrinoma is due to excessive gastrin secretion that leads to production of HCl by the parietal cells. The elevated gastrin, the key here is the elevated gastrin confirms our diagnosis. We have another case scenario here. A 60-year-old diabetic patient complains of early satiety. So we have a elderly diabetic who complains of early satiety, bloating and constipation. So he doesn't like to eat food. He's always bloated. Abdominal distension is there. Constipation is also there. Gastric emptying studies show delayed gastric. His constipation is because of this delayed gastric emptying. It is a delayed gastric emptying that has caused constipation. Now, which hormone deficiency is most likely contributing to this symptom? Okay. What happens here is in aged diabetics, okay, most of the time that is gastroparesis is present, and the most important cause of gastroparesis is deficiency of which hormone? Motilin. That is why he is having early satiety, bloating, constipation. Why? Because the migratory motor complexes are not there. So during the interdigestive phase, the food particles from the previous meal is not getting washed away. It's not getting swept away. So he is not able to eat his next meal. And these products that are remaining is causing bloating, and decreased gastric emptying is causing him constipation. Okay. So this is a direct answer. Answer is Motilin. As I told you, diabetics usually have gastroparesis, which is due to impaired Motilin function. Diabetics have gastroparesis, which is associated with impaired Motilin function. Okay. Motilin regulates the migratory motor complexes and promotes gastric motility. A deficiency of this is the reason for the symptoms like bloating, constipation and decreased satiety. Now, here we have another patient who undergoes extensive small bowel recession. So, the small intestine is surgically removed. Post-operatively, he develops severe diarrhea and malabsorption. The small intestine is removed. As a consequence of that, he has severe diarrhea and malabsorption. Which gastrointestinal hormone is most likely deficient? So, this patient, what they are asking is, when the small intestine is removed, what is the hormone which is causing this type of diarrhea and malabsorption? What is the answer here? It is peptide YY. Gastrin you can already rule out because it is from the stomach. Why not secretin and cholecystokinin? Okay, secretin and cholecystokinin, what do? They actually inhibit the gastric motility. It is the peptide YY which is actually secreted by the ileum and colon in response to food intake. So here we have another patient who undergoes extensive small bowel resection. Post-operatively, he develops severe diarrhea and malabsorption. So which among these hormones is most likely deficient? Okay. So when you look at all these hormones, gastrin, secretin, cholecystokinin and peptide YY, the peptide YY is the hormone which regulates the movement of your ileum and colon. So this peptide YY, because of the small bowel resection, the cells producing peptide YY is lost. So this leads to unregulated gastric emptying and diarrhea. So thus, to summarize, I would just want to just emphasize again, the gastric glands as well as the crypts of Lieberkühn are those specialized region where we have these enteroendocrine cells which secretes different types of GI hormone. To remember, goblet cell secretes mucus which protects the stomach lining. Parietal cell secretes gastric acid, important for digestion. Chief cells secrete pepsinogen, which is important for protein digestion. The D cells secrete somatostatin, which inhibits acid secretion, and G cells which produces gastrin, which stimulates the parietal cells to secrete gastric acid. Hope you understood this concept. Thank you.